It is 6 p.m. here on the west coast of the United States, so we are going to get started with our webinar. So thanks everyone for joining us. Today's webinar is called Made in the Shade, How 11 Research Studies Cleverly Use Canopy Analysis to Measure Crop and Forest Health. So today we will talk about our CI 110 plant canopy imager and how it's been used in various research applications. But before we get started, let me advance my slide here and want to introduce to you our webinar moderator. Jamie Fuller is our marketing coordinator, and she's our newest member of our team here at CID Bioscience. So she'll be taking the place of Susie as our moderator, and she'll be looking at the chat box as well as the Q&A boxes and monitoring those as we go through the presentation. So just some housekeeping before we get started. For all questions that you might have, please use the Q&A function in Zoom. So in this image here on the right, you can see that the chat box is highlighted in orange and the Q&A box is pointed to with that green arrow there on the right. So if you have any questions related to the content of this webinar, related to our products, anything like that, please only use the Q&A function so that at the end of the webinar, I can go and address those questions one at a time. Please do not use the chat function, which is the function that's highlighted in orange in that graphic there. Don't use that chat unless you are experiencing technical difficulties like a lack of sound or video. Please, like I said, put all those questions that you might have into that Q&A function and leave the chat for any sort of technical issues. But Jamie will be posting all of our relevant links, so links to these studies, other links that might be found within this webinar. Those will be posted in the chat. So do check the chat box. Just please, if it's a question related to the content, use that Q&A function. Just to introduce myself, in case you haven't spoken to me before or are familiar with me, my name is Galen. I'm an application scientist here at CID Bioscience. I have a bachelor's degree in biochemistry and a master's degree in food science from Michigan State University. I'm an IFT certified food scientist, and previously, before joining the CID Bioscience team, I managed an analytical food testing laboratory and also worked as a food safety consultant for the food manufacturing, produce, and cannabis industries. Our other application scientist who you might be familiar with, he presented this webinar this morning. His name is Eric Munoz Garcia. He's got a BS in biohealth sciences from Oregon State University. He's worked as a chemist and a DNA analyst in the dietary supplement and natural products industry. He's got a lot of experience in testing, identity testing, and product specification testing, food label compliance, and stuff like that. So he's a great source of information as well. If you have any technical questions, him and I are the ones that you will want to address those to. So let me lay out our agenda for you today of what this webinar is going to look like. So we'll go over, we went over the introductions already. We'll kind of go over a CI 110 product overview with some features and some specifications. And then we're going to jump right into the current research and talk about who's using the 110 and how they're utilizing it. And so we'll show 9 different studies that we've handpicked to kind of display the variety of research fields that the CI 110 can benefit. And then we have 2 guest speakers today that will be talking to us. Unfortunately, these will be recordings for you guys. They were present live for the morning webinar, but we do have the video recordings of their presentations, which I will be showing to you later on in this webinar. And so Derek Godwin, who's a extension professor at Oregon State University, and also Brendon Anthony, who's a current PhD candidate and has done some really good research previously at his master's at Washington State University that he's going to talk about. And then we'll end this webinar with our Q&A session where you can ask questions, and I will do my best to answer them. Depending on our time constraints, we might have to have you email me personally, and so that we can get back to you if your questions of a very technical nature. It might be better for us to communicate that over email so that we don't take up the entire question and answer session trying to answer one question for you. So. All right, let's hop right into it. So our company, CID Bioscience, has more than 30 years of experience in plant research and instrumentation. So, our mission is to serve the research community and any commercial needs in the agriculture sector, in the forestry sector, ecology, and many more. You'll see there's a lot of different applications for the CI 110. And what we, really take pride in is the fact that our instruments are non-destructive measurement tools. We help researchers acquire consistent and high-quality data. Our data is, instantly acquired, really accurate results. Our instruments are durable and portable for the field, and we always pride ourselves on our data transparency. And all of our product lines are engineered, tested, and manufactured under the same roof here at our headquarters in Camas, Washington. So that's something that we also really take pride in. So talking about the CI 110, the CI 110 is a plant canopy imager, and it utilizes a hemispherical camera so to do hemispherical canopy photography, as well as image analysis with light measurements to non-destructively calculate leaf area index, as well as a variety of other canopy parameters. It has a self-leveling digital camera that does 150-degree images of the plant canopies. And also the arm of the instrument is lined with 24 photosynthetically active radiation sensors, or PAR sensors, which can be used as an alternative measurement of LAI and to assess current radiation levels at the site, and also to evaluate sunflex. So our newest updated design is an all-in-one device, as opposed to a separate wand and computer unit. And this updated ergonomic design has a 7-inch touchscreen and a trigger that allows you to see either live views and or a image after you've released a trigger actually captures the image to give you these nice crisp images for further analysis. And it also has the ability to add or exchange filters over the camera lens to ensure that you're getting the most contrast between the sky and your canopy, and to ensure that your data is of the highest quality possible. So some features of the instrument, as I've already said, I think the number one feature that most people find useful is that this is an all-in-one unit. So it's a convenient all-in-one unit. It's easy to use. And I can actually show it to you here really quick. I can pull it out. And this is, if you can see, the CI 110 plant canopy imager. And it is a really easy ergonomic design. It's all in one. And so it can do all the calculations for LAI of your plant canopies across multiple size classes. The camera lens has an adjustable focus. So for canopies that have varying heights, it works really great. The calculation of the gap fraction distribution, the leaf angle, and the plant canopy extinction coefficients all happen simultaneously. It does not require an above canopy reference reading, which is another benefit. So you don't have to worry about finding a clearing, or you don't have to worry about having a second sensor somewhere out in the clearing to get that above canopy reference. And then some other features are that, you can perform these measurements pretty much under any sky condition. And when you take these measurements, you instantly are able to see on the screen your location that's acquired via 4 different methods due to our amplified antenna. And the internal compass is great for standardizing your measurements across locations. And as I mentioned, the filters that are available to optimize your images across varying light conditions is another benefit of the instrument. So we think this is a really drastic upgrade from our previous model, and we're really happy with it. And we've had a lot of good success stories with people using our all-in-one CI 110 unit. So let's jump right into some of these applications that I was talking about. As I mentioned, there's really almost limitless applications for this Canopy imager. So you have your standard forestry, forestry management and conservation, repair, investigating canopy densities in any sort of typical forest situation. But we also have people using it in agriculture. So using it for field and orchard management, or looking at various soil or plant treatments and their efficacy. And then also people using it to investigate things like plant biodiversity and distribution, looking at biomass production. And then we even have users that are using it for some pretty non-conventional uses, like urban planning and development, or architecture, civil engineering applications. So it really isn't limited to just the forestry application, which might be the first application that comes to mind when you think about investigating plant canopies. So hopefully the next 9 studies that I show you, and then the following 2 studies from our 2 guest speakers will help to kind of show that this instrument has a really wide range of applications. So let's dive right in to our first study that we picked. And this is a study that was conducted in India, and it was looking at the effect of different tree spacings and also investigating poultry manure and compost effects on the growth and yield of bell pepper under this agri-silviculture system. So instead of just growing bell peppers out in the open, they were growing these bell peppers, underneath and in conjunction with and next to these Grewia aptiva trees. So what they used our instrument for, and you can see the excerpt on the right is a direct quote from their paper. So they essentially were using our instrument to investigate the parameters like leaf area index and photosynthetically active radiation in order to help determine the effect of this tree spacing. So how far apart these trees were planted influenced the growth of the bell pepper, and the yield of bell pepper. So looking at the changes in LAI, if you look at the table, they're showing the different tree spacings and the associated PAR levels and LAI levels, and kind of associating that data to the eventual yield and growth of the bell pepper to determine which of these tree spacings is the optimal tree spacing to have in order to get the highest growth and yield of bell peppers. So that is, one use in an agriculture kind of application to investigate, different treatments and different methods of tree spacing to look at basically crop yield. So our next study was looking at actual kind of mitigation strategies to control and kind of rejuvenate sweet orange trees that are affected by HLB, which is a disease that causes the roots to actually kind of diminish. And so the thought behind this study was that if they'd actually went and pruned the canopy of the tree, that and that therefore reestablishing a kind of normal ratio between the root structure, which had diminished because of the disease, and the size of the canopy. So by pruning, they could reestablish that ratio to a normal level, so that there isn't as much stress from like a stress carbohydrate cycle. Then that would actually kind of reinvigorate and rejuvenate the plant or the trees. And so they used the CI-110 to look at the leaf area index as a estimate for canopy density, in order to determine how much canopy density to eliminate in their studies. So they did, I think, 4 different treatments. They did a 50% or a 20% or 0% reduction, 25% reduction, 50% and 80% reduction of that canopy. So they, had to make sure that they had precise measurements of their canopies, of their basically using the leaf area index from the CI-110 in order to control that variable as they pruned, to make sure that the treatments were consistent across all the different treatment groups. So the next study that we picked was looking at another kind of agriculture type situation where actually they were essentially looking at finding a way to use hemispherical photography to estimate things like the biomass production and the yield, as well as the LAI of these crops. And so this study wanted to be able to use, and as a non-destructive method, hemispherical photography, which is what the CI-110 uses, in order to kind of estimate in these large fields of cotton, and estimate what the biomass production will be, the LAI, and the irrigate or the yield of this irrigated cotton. And so they did it with basically it's kind of trying to adopt a new technology, which is this hemispherical photography, and then kind of evaluating it against other methods, more destructive methods. So what these graphs show in the bottom right are actually plots of the LAI that was taken with the hemispherical photography against a different method, like a destructive leaf area meter. So using the Lycor LI3100C, they actually, after measuring with the CI-110, they went and removed all the leaves and ran them through the leaf area meter from Lycor. And then they also did measurements, classical measurements based on the midrib length of all the leaves. And so comparing those methods, basically trying to assess the quality of the hemispherical photos versus the classical destructive methods, and trying to find a way to make it a more convenient way for them to estimate the LAI and the biomass. And they found that it actually was a very good method to do the estimation of LAI and biomass production. And they also mentioned at the end of their paper that it looked very promising to actually estimate yield, but they mentioned they have to do more research in order to kind of confirm that it would be a good method for estimating yield. So the next study that we looked at is a kind of more of a classical forestry application of just looking at plant spacing in a poplar plantation. And so looking at how the plant spacing influences the canopy structure, the biomass production, and other properties like the stem roundness of these poplar trees, and trying to basically figure out, the best spacing or the optimal spacing, the most economical spacing to give the best yield of biomass from the trees. And so you can see these are some pictures that they took with the camera, these hemispherical pictures with the CI-110. And obviously, in these kind of situations, it was very, the CI-110 was more of the prominent tool used, because it's a very important application for looking at leaf area index, leaf angle, and par levels. And so essentially what they did is, use our CI-110 and used it to, take images, 1 meter above the surface soil. And then they evaluated the canopy structure, indicators like LAI and leaf angle and the transmission coefficient for the photosynthetically active radiation, and compiled all that data to kind of determine which under which conditions these trees actually thrive. So that was another kind of great kind of standard forestry application that we would kind of expect our instrument to be very useful in. So the fifth study that we looked at kind of also is a forestry application, but also on the ecology side of things. So essentially, this study was actually investigating how after a typhoon, Haiyan, I believe it was, in the Philippines, after it hit, there was a reforest that they actually were already planning on looking at how that forest was influencing the water retention and streams and things like that. Actually, it was kind of more of a hydrology, experiment. But then what it kind of transformed into was looking at how this typhoon influenced all those conditions, and it is influencing the rainfall that is actually being accumulated into streams, because a lot of these forest canopies, after the typhoon had hit, became, had a less LAI. They had been partially destroyed. And so what they used the CI-110 to do was that they were looking at the canopies basically over time, over these years. So basically before, when there was a full reforest, immediately after the typhoon, when it was mostly destroyed, and then looking at it, after it had time, about a year to grow back. I believe this was a 3-year study. And so essentially what they looked at with the CI-110 was looking at the plant canopy and how the leaf area index changed over those times in the areas where they were actually, it was in the same areas where they were collecting the rainfall or the throughfall of rain through those canopies. And so these graphs just show the changes in the plant and canopy size over those times, over the course of this investigation of this reforest. So again, a great tool to use to accurately assess the LAI and the canopy size and density, and can be used in applications beyond just looking at tree spacing. Now you can use it as a tool to look at recovery from natural disasters and stuff like that. So I thought this is a really interesting study. The sixth study that we investigated was looking at actually, as the study 2 studies ago, when I was mentioning that they were looking at the hemispherical photography as the new technology, and they were kind of evaluating it against these traditional destructive methods. This study takes it a step further, and actually they were trying to basically utilize images from the Sentinel-2 satellite and find a way to actually get accurate vegetative index readings simply from the Sentinel-2 images that they were getting. And so they were using the CI-110 as kind of their reference method, and then comparing that and using that to create these relationships or regressions between the LAI and various vegetative indexes like the NDI and NDVI. We're hoping to create a model that could predict leaf area index and chlorophyll content, and then hopefully through those two parameters also be able to predict these vegetative indices, all just from this satellite image. So in this case, it was the CI-110 that was being used as the reference method for LAI, as opposed to the previous study that I mentioned where the CI-110 was being used as the experimental method, and the destructive methods were the reference method. So I thought this was a good kind of full circle of technology example. The next study was looking at basically the transitions between the forests and savannas, because these are very actual abrupt changes between savanna and forests. Something I learned while investigating this study is that actually the transition between a savanna grassland that is really susceptible to fire and a dense mature forest can sometimes be a matter of meters. These two kind of separate mosaics exist with this very strong divide between the two, but there is some medium ground, and so essentially they were using the 110 to look at how the canopies from these from the transition from the mature forest to the the thicket forest kind of margin, the in-between space, and then the savanna, how that change in canopy cover and canopy density actually influences fire susceptibility and things of that nature. So the LA or the CI-110 was used to look at the LAI in this case, in all of these different sampling regions, as shown in the image there on the top, as all those cylinders show a sampling area where they investigated how much canopy cover there was, how much photosynthetically active radiation was getting through and allowing grass to grow, grass that would actually be fuel for these fires. So a really interesting study that looking at and kind of summarizing and explaining these really intense, basically divides, these two mutually exclusive mosaics that like exist right next to each other. It was a really pretty cool eye-opening study. So another great use of the CI-110 in this study. Our eighth study is another kind of deviation from the standard investigation using the CI-110. And so these researchers at the University of Michigan were actually looking at how and basically kind of trying to describe exactly what situations can cause this dispersal of exotic plants through what they call seed rain coming from birds. And so looking at these different types of fruit-bearing trees, both native and non-native exotic species of these trees. And so how they actually used the CI-110 in this situation was to not necessarily for any sort of data that they would use in their final evaluation or assessment, but it was actually more to make sure that their variables were being controlled. So they actually utilized it to make sure there was no significant difference in their gap fractions among the plots that they were looking at when they were doing this study. So they wanted to, control those variables. So I thought it was a really cool use of the CI-110, to really make sure that their data was as robust as possible, that there were no confounding variables. They actually used the 110 to eliminate the chance of there being influence from different canopy covers and different densities of canopies within these forests where they were investigating the seed dispersal and the spread of these exotic plants. And then the last one that I'm going to talk about before I end up handing this over to the videos of our guest speakers is another really cool kind of ecology-based application where these researchers were essentially looking at factors that are influencing the biodiversity of these butterflies in northern Mexico. And so this landscape is a secondary succession landscape, and they were looking at, you know, across different dry and wet seasons and across, you know, different variables such as leaf area index and photosynthetically active radiation, looking at what are the, you know, variables that are influencing the biodiversity of these butterflies in this particular type of landscape. And so actually they didn't find any sort of significant correlation between the leaf area index and the photosynthetically active radiation and the biodiversity of these butterflies, but it was still, you know, a very valuable data to learn pardon me, you know, what factors were indeed influencing the biodiversity of these butterflies. So we're gonna kind of shift. We're still talking about, you know, applications and research, but we're gonna kind of focus on three different people. The first is John Eames. He's a research biologist for the US EPA, and they use the 110 CI 110 Canopy imager to quantify forest biomass, looking at air surface exchange processes and speciated deposition budgets of nitrogen and sulfur compounds in key ecosystems. So they do a lot of really cool work with the CI 110 and look at multiple forests across the Appalachian Mountains and North Carolina. And so they use it in forests that have extremely tall trees and tall canopies. So they basically take the LAI readings in these really tall forests every 5 meters from the bottom to the top of the canopy. So that's really cool application, and as you can see there, it looks like he gets to also do some cool aerial imaging stuff as well. So what I'm going to do now is introduce our first guest speaker, who is Derek Godwin, who is a professor at an extension faculty really at Oregon State University. He's in the biological and ecological engineering field in the College of Agricultural Sciences. And so what I'm going to do is play a video of him giving his presentation earlier at today's earlier webinar. And then once that's done, I will move on to our next guest speaker. So bear with me for a second while I switch to the video, and then we'll get that playing for you. Make sure this is. Pardon me 1 second while I get the sound. On for you guys. All right.
Good. Looks like you've been on my Facebook. So I'm Derek Godwin, and I'm an extension faculty working for Oregon State University. My home department is in the biological and ecological engineering department there in the College of Ag Sciences. So I will, there we go. Okay. So yeah, I'll let Jamie know when to change the slides here. And so anyway, what I want to share is that some watershed riparian systems research that I'm working on with Dr. Carlos Ochoa. He heads up the eco-hydrology lab in the Department of Animal and Rangeland Sciences. As I mentioned, he has the research component or research assignment, and my assignment is extension faculty. And so this research that we're doing, I'm pretty new to this. So I'm kind of sharing some ideas and initial work that we have. So Dr. Ochoa has studies in both Oregon and northern Mexico. And so what I'm showing here on this slide is that we do our research in working landscapes. So they're all a working agriculture landscape, but with different ecosystems, and each of them involve commercial producers. So this is giving you an idea of, in Oregon, the 3 sites that we have and the different ecosystems that they're based in, and then also the northern Mexico site. So what I'm mostly working in is a project in Western Oregon in the Willamette Valley on an Oak Creek watershed. And within this watershed, we have a variety of monitoring locations throughout the watershed, looking at everything from soils and soil moisture to vegetation cover to a variety of water quality parameters. But the kind of the focus of the study that I'm talking about is this, as you see in the watershed here, we have monitoring stations for stream temperature from the headwater area all the way down to the mouth. And I've separated out a couple of reaches here, one being the Sheep Center reach here that's a piece of OSU property that's grazed with sheep, and then also have a reach down here that's at our dairy. So it's there's some livestock or dairy cows grazing that area as well. So what I've done is to zero in on a couple of reaches to focus on stream temperature. One being the upper sheep site, and that's about it's about 600 or so meters, and the dairy site is around, it's about 1,200 meters long. And both of these areas have been under a conservation riparian enhancement program project. So essentially they have been fenced and planted, and has been managing with the riparian buffer for about 15 years or so. The sheep side buffer is about 15 to 25 feet wide on both sides, and the dairy one ranges from about 25 feet over to a little bit over 100 in some areas. And so what I'm looking at is that this watershed, this stream, is listed as water quality limited in terms of stream temperature. And so what I'm looking at is getting to better understand the various factors affecting stream temperature in these 2 reaches. So with the Canopy imager, what I've done is in those reaches, I've created transects and taken pictures, essentially images, every 10 meters as I move through the main part of the channel. So what I'm trying to get is canopy density above the stream. And so I take these pictures at about 1.5, 2 feet just above the water level, essentially as low as we can get, and then do this up and down the reaches to get a pretty accurate sample. So as I mentioned, in this case, it has pretty good cover on both of these stream reaches. So what I'm also showing here is the picture, as well as the filter that I'm using, the blue filter, in order to try to separate out the leaf and canopy from the sky, and then the final picture here. Okay, so in that first one, looking at canopy cover and using that as a component for stream temperature modeling. A second part of our research, we have a different student, Nicole Durfee, that is kind of a specialist in UAVs, and she's using these for a variety of different kinds of analysis, one being the leaf area index. And so what we thought is that we would look at the canopy imager and compare the Canopy imager with the aerial photos that she's using in a few different environments, and just kind of compare the two different methods. And obviously, in some cases, the flights, the UAVs are much easier to use, but in other cases, the Canopy imager would be easier to use as far as covering the area. So that's that one. One more. Okay, and so this one, so it's back at the Sheep Center in Oak Creek, and we have a plot study there that's in a wetland. And so we've set up a grid there and done a variety of samples. But in relation to the Canopy imager, what we're looking at is, again, this comparison of using the Canopy imager from the ground up and looking at cover of the vegetation.
All right. So that was the end of the video for Derek's recording. It looks like he got cut off a little bit at the end there, but we will move forward with the next guest speaker that we have. Let me advance through Derek's slides here. And the next guest speaker that I'd like to present is Brendon Anthony. He is a PhD candidate right now in horticulture at Colorado State University. He previously was a student at Washington State University, where he did his master's in horticulture. And I believe his presentation will look at some research that he did while he was there at Washington State University. So bear with me for 1 second again, and I will get that video loaded up for us.
So yeah, my name is Brendon Anthony. I'm a PhD candidate here at Colorado State University, and I'm just going to be sharing a little bit about the research I did with the CID 110 over at Washington State University, which was a part of my master's thesis. So we can begin. All right, so yeah, just as I said, basically what we were trying to do with the CI 110 is really try to understand how, large or how, small our canopies are in the context of a high density apple orchard. We wanted to try and optimize the methodology for this tool so that we could use it to inform certain horticultural decisions. As you can see, the model we were using is not the updated version. So just I'm sure there's some differences that Eric and the rest of the team can talk about between the older model and the newer model, but just wanted to preface that this was done in 2016, so this was the older model. So just to give some background on why leaf area index is important, again, just to define the parameter, it's the ratio between the leaf area and the ground area. And it's important because it allows us to assess, the health and vigor of our trees. It correlates well with photosynthetic efficiency, which at the end of the day, it's all going to be generating photosynthates for our fruit production. As we see, as the canopy expands, you're able to intercept more light. You're able to intercept more light. You can then have higher quality fruit and increase your orchard productivity. So understanding the canopy expansion, how large your canopies are, is a really fundamental thing for especially tree fruit growers. And being able to manage your canopy is really important, especially in a high density context where you have the potential to shade your fruit and then reduce the quality. So it's really important for us to be able to quantify and manage the canopy properly. So just some background again on why this indirect quantification method is so important. Because as you guys have all been saying, allows for real-time estimation. Doing direct quantification and taking all the leaves off and scanning them takes a lot of time. I did it for part of my master's, so I know firsthand that it's not the most fun task to do. So being able to just, snap a photo and get an estimation is, obviously a huge benefit. But we wanted to see, how accurate is this, and is the estimation going to correlate well with the actual destructive measurements. So there's really no direct standardization in terms of indirect leaf area index measurement methodology. This has been reviewed in a lot of different review papers, but it doesn't seem to be a consensus. A lot of the tools, you have to take the photo with a camera and then use a different software. So really the huge advantage of this tool is really this all-in-one package where you can instantly get your leaf area index in the field on the same tool with the screen and the imager. So it's really all-in-one package. And at the time of the publication, there really wasn't a lot of published data on this. Obviously, you guys have seen over the course of the presentation, there's a lot more papers that are using this tool now. But when we were developing this methodology, it hadn't been used in an orchard setting yet, just primarily used in more ecological systems. So our objective, again, at the end of the day, we just really want to be able to inform our horticultural decisions. So we wanted to develop an optimal methodology to yield accurate results on the leaf area index in our high density orchard context. And we also then did a destructive analysis to be able to compare with estimations so we can see how close we were getting to the real values. So some of the aspects that we were looking at were the light environment under which the images were being taken. So we created artificial light environments using different shade nets, as well as having a no netted control. And then we measured at different different heights from the base of the orchard, going from 10 centimeters to 40 centimeters, which is basically at the initiation of our canopy. So we got pretty close. And then using 2 of the different automatic thresholds embedded on the tablet there. So we did this across 3 triplets of 9 trees, so 3 groups of 3 trees, and we measured this under uniform sky conditions. And the data that I'll be showing is expressed as delta LAI, so the difference between the destructive versus the estimation. And so the smaller the delta LAI, the more accurate the result. So we'll get into some data here. So first off, we'll see the impact of the netting or the artificial light environments on the ability to accurately assess leaf area index. What we noticed is that when we put the blue nets over, they created a very dark environment. And when you have a very dark environment, it's difficult for the tool to discriminate between parts of the tree that are wood versus leaf. And so in that case, when it was a really dark environment, you just got the totality of the tree, wood, fruit, leaf, and so then your leaf area index goes up artificially. Then the problem in the inverse side is if you have a really high light environment, and you can see that here, especially on this top image where we had the white net reflecting the light, that then creates this kind of leaf washout effect. And then you're artificially deflating the accuracy of all the leaf area there. So what we decided and the results showed is that with the no net and just our normal light environment, an earlier morning where you have some light but not too much, that yielded the most accurate results for using the Canopy imager. When we look at the impact of height on accurately estimating leaf area index, we realize that the further away you are from the canopy, the more accurate you're going to be. So lower to the ground, you're able to get the totality of the canopy, whereas as you get closer and closer, you are essentially reducing your ground area, or in the context of the camera pointing up, your sky area. And so in that essence, if you are reducing your ground area, then your overall leaf area index is going to be inflated again artificially. And so I think this is why this tool is used so well in very large trees, like in a forestry context, because, when you're standing at the base of the forest floor, you have a bigger picture of the canopy. And in the context of an orchard where that canopy is initiating at 50 centimeters, you get too close, you're going to have some accurate representation. So for us, the further away from the canopy you were, the more accurate estimations. You can see that here listed in the figure. So lastly, just looking at the different thresholds that the software has, and I don't know if the software has been updated with different thresholds, but these are the ones that we had at the time, looking at Otsu and entropy. Otsu did a better job of delineating again between leaf and wood, and so allowed us to have a better accurate estimation of the leaf area index. The entropy is a little bit higher saturation threshold, and so again, you have that tendency to artificially inflate the true leaf area index. So what we did then is compiled all of these together, and we kind of got a standard optimization methodology where we did a 3-way ANOVA. You can see all the different combinations that we tested, and the one highlighted here in green, again, compiling all of the results I just showed. The no net, further away from the canopy at 10 centimeters, and then processed with the ATSU. That got you a difference in LAI of about 0.19. The margin of error listed in the literature is about 0.25. So this kind of got us within that zone where you can get a fairly quick and fairly accurate estimation of leaf area index. Again, this is ideal to do across multiple trees, especially in a high density orchard context where you have a lot of interference from neighboring trees. And so it's difficult to just measure a single tree because you have trees planted so close together. You're now starting to estimate leaf area coming from them. So it was easier for us to group them together. And when we did, we had more accurate results. Again, you want to have a nice optimal balance of your light conditions. So this was around or less than 500 par in the sky. If you have too high of light, you have interference with your camera lens. You can have sun flecks and washing out your leaf area. But again, if it's too dark, then you can't necessarily delineate between the foliage and the wood. And so lastly, our conclusion here is that it provides enough accuracy to be able to inform management techniques. So for example, if we were to estimate our canopy and see that the leaf area index is too high, then we can say, hey, okay, let's go out and do some summer pruning, reduce that canopy so we can make sure that we're having optimal light interception on our fruit so we can have high fruit quality and better coloration. So that's all I have. If you guys have any questions, feel free to shoot me an email. This is a publication listed here. I'm sure they'll provide the link if you guys want to learn more about the details in terms of the methodology that we developed. Thanks, guys.
Really, the point that we want to drive home today is that the CI 110 isn't just limited to the typical forestry application. It really can be used in such a wide variety of fields of research, agriculture, in that orchard setting that Brendon used it, hydrology and ecology, like how Derek was using it, and many more. And just because there might not be publications on it, as Brendon had experience with, there might not have been a publication on that, on the use of it in an orchard setting, but it really is still a useful tool to be able to use. And so if you think that it's something that could be valuable to look at, to be able to measure the leaf area index and the photosynthetically active radiation, your canopy densities, then really it can be a great tool for you. And so, as I said, anyone that's a researcher in those fields, in the fields of agriculture, forestry, ecology, urban developers, architects, really everyone that is currently trying to use kind of an outdated system and a destructive method of measuring LAI can benefit from using our CI 110 because it's this all-in-one inclusive tool. It's non-destructive, provides instantaneous results. And so if you have, as part of your research, or if it's a graduate student, that has been, hitting their head against a wall, having to destructively measure leaf area index, this is a great tool to speed up that process. And then just kind of going back to the features of it, like I said, it's just this all-in-one ergonomic tool. It's non-destructive. Combines the hemispherical photography and the image analysis. And it gives you instantaneous acquisition and analysis of your data in under most conditions. So really, it's a great tool to use, and I look forward to seeing all the great new research in 2020 that comes out that utilizes our instrument. If you have any questions, if you already own an instrument and you have a support question, please forward that on to our support team. The email is support@cid-inc.com. And otherwise, if you are curious about the instrument and curious about how your application might benefit from the use of a CI 110, please feel free to reach out to myself or Eric Munoz Garcia, our other application scientist. You can reach out to us personally. and email us, and we will be sure to get back to you and figure out how the 110 can benefit you and your application. So just want to really quick let you guys be be aware of some upcoming releases we have. We do have a new leaf spectrometer, the CI 710. It is a vast improvement, similar to the 110's evolution. The 710 is evolving from a system that requires two hands to a kind of a single-handed unit. It's an all-inclusive, all-in-one instrument. Again, be able to measure reflectance, absorbance, and transmittance. Tons of built-in indices for your vegetative indices like chlorophyll and anthocyanins and carotenoids. And additionally, the ability to use partial least squares regression modeling and compatibility with our fruit maps harvest tracking softwares. Will all be standard in the 710. It's a vast improvement from our previous model, so we're really looking forward to releasing that in this upcoming year. And then also our F751 Kiwi quality meter from Felix Instruments. We'll be releasing that in the coming month or two, and that will allow you to instantly and non-destructively assess your Kiwi quality and maturity for looking at your indices like dry matter and bricks. So those two products, be on the lookout to stay updated on those releases and any other future releases we have. Please follow us on social media. You can find us on Twitter and Facebook and LinkedIn. Also, our website is a great tool. We regularly release newsletters and blog posts. And of course, if you have sales questions, feel free to reach out to us via phone or email us at sales@cid-inc.com. And if you would like to request a quote, we have this link here. Obviously, you won't be able to click on it on this presentation, but Jamie will post it in the chat function, and you can follow that link to request your quote for a CI 110. So I think we'll move on now to our Q&A, and I will open up the Q&A box here. And let me just make sure quick first that there's no questions in the chat box. All right, so great. All right, so the first question is, can you explain how canopy density relates to photosynthetically active radiation? Okay, so if you think about it as the denser the canopy, the less light you're going to get through. The same way it works with photosynthetically active radiation. So if you have a very dense canopy, the foliage underneath and the vegetation underneath that canopy will be receiving less of that photosynthetically active radiation than if you had a more sparse canopy where there was more gaps in your canopy that allows unrestricted sunlight to go through. And so even with, if you have a tree with very transparent or translucent leaves, those leaves are still, in that canopy, are still absorbing a lot of that photosynthetically active radiation. So even though it might appear that you actually, the environment underneath the canopy is bright, light, like very well lit, you still will have reduced photosynthetically active radiation reaching that vegetation underneath. All right, and our next question is, what is the battery backup, especially for forest applications where the recharging may be a problem, any solar charging facility? So our solution to that, and with all of our instruments, our solution to that is we actually use rechargeable lithium ion batteries as our power source. So the instrument itself doesn't need to be recharged. You just need to carry extra sets of charged lithium ion batteries. And those can be inserted into the instrument. Once your initial set dies, you put your freshly charged set in and you're good to go in the field with no gaps in between, having to go take a break to recharge or anything. So actually all it takes is a few extra lithium ion batteries and you're good for an entire day. Our battery charger can be used in the field with solar panels as well. So if that is something, if you're out for extended periods of time and you don't have access to a facility with electrical outlets, you can hook it up to a solar panel and then charge your batteries that way. If you run out of charged batteries to use and you're out in the field for extended periods of time without access to electricity. And also, the battery life is exponentially better on on our new CI 110 model. So really, your batteries shouldn't be dying that often that you need to take breaks throughout the day to recharge your instruments, or to recharge your batteries rather. The next is I see a few studies mention taking readings early in the morning or late evening. What will happen if readings are taken at noon? So really, people, as Brendon mentioned in his discussion, the reason why people tend to take readings either in the morning or late evening is to get the optimal level of lighting to get the most accurate LAI reading. So if you are taking a measurement at noon when it is extremely bright and your canopy is fairly sparse or not completely blocking out the light, then what's going to happen is you're going to likely have altered LAI readings due to the fact that some of the leaves might get washed out from the reading. As Brendon's data showed, when their light was too bright, they were getting some washout, so their LAI was actually being under predicted. So that's why taking them in the early morning or the late evening might be the best option to get the most accurate LAI reading so that you have the best contrast between the sky and your canopy without any sort of interference from it being too dark or too light. So the last question here is there any other language support other than English for the user interface? And let me quick ask our engineer that question. So we don't have anything other than English for the user interface at the moment, but feel free to email us and let us know if that's something that we should be looking into adding. We would be glad to look into that for you. Yeah, it's a pretty easy process. Our engineer just encouraged me that it is a fairly easy process to get other language support into the user interface. So if that's something that you would need, then please let us know, and we can work on that for you. All right. If there are no other questions at the moment, then I just want to say thank you again for joining us. And please, if you have any questions, further questions that weren't answered in today's webinar, just go ahead and email myself or Eric, and we will be happy to answer those questions. If you have support questions, you can email our support portal. If you have pricing questions, you can email our sales team. And then if you want to just request a quote, feel free to click that tiny URL link. And I appreciate you guys joining us, and I look forward to hearing from some of you about your future research with the CI 110. Have a good day.